Hey, good afternoon, everybody. We're so excited to have you here today. Today we're going to be talking about advancing data collaboration in rare disease and epilepsy research. I'm Sam Volchenboum. I run Data for the Common Good at the University of Chicago. I'm a pediatric oncologist, and I'm so excited to tell you about the work that we're doing and to have several special guests here with me. A few notes of logistics. This meeting is being recorded. We will send out a link to attendees in the coming days so that you can watch it and hopefully share it with other folks that might be interested. At the end of every speaker's presentation, we'll reserve a few minutes for a Q&A, and you can utilize the Q&A box to submit questions to presenters. For any technical issues going on, just email Bailey, who has been the mastermind of putting this together, and her email address is there. And I wanted to point out that the link to this presentation that I'm giving is at the bottom of every slide, and you should feel free to download it. You're welcome to do that at any point, and feel free to share it, and we'll put this link into the chat as well. So what we're going to go through today is listed here. I'm going to spend a few minutes talking about our group, Data for the Common Good, and then I'm going to transition right into our collaboration with the Chan Zuckerberg initiative on our monogenic epilepsy project. When I'm finished speaking, we're going to have a talk by Justin West, who's from the KCNT1 Foundation, and then we'll have a short break, and then we'll come back and we'll have a talk on our work with EEG biomarker discovery by Doug Nordli. And then we'll have a talk by Larry Hunter on his new project with CZI about language model research in epilepsy. And then we'll have a brief panel discussion where we'll have a chance to, again, answer questions from you all. And then at the top of the second hour, at the end of the second hour, we're going to be joined by Dr. Rob Califf, the FDA Commissioner. He's going to make some remarks, and he should be able to stick around for a brief discussion afterwards. So I'm so excited to tell you about Data for the Common Good. And I think it would make sense to start with some lessons from childhood cancer. So this here shows the survival rate for pediatric acute leukemia over the past 70 years or so. And this article had an interesting quote. It said that a diagnosis of childhood leukemia in 1950 was uniformly fatal with an average period of 3 months. And basically, there were no therapies. We had some steroids, but other than that, there was really no way to effectively treat any child with leukemia. What happened over the past 70 years with pediatric cancer was the formation of cooperative groups to study cancer. And on the left is a little tree showing how we got to the current Children's Oncology Group, which formed in 2000. But it was made up of many years of other groups coming together to perform studies for pediatric cancer. On the right is a map showing most of the Children's Oncology Group centers in the United States. Worldwide, there's about 250 institutions that are part of the Children's Oncology Group. And I make this point because it's so critical for rare diseases that we have groups like this that can bring data together for study. And for pediatric leukemia, here's what's happened. The curve on the bottom in pink is looking at the survival rate for pediatric acute leukemia in the late 60s. And you can see that after 5 to 10 years, less than 10% of kids have survived. And over the past 60 years or so, these survival rates have gone way up, to the point that now a 5-year survival rate for children with ALL is well over 90% for standard risk disease. Just think about that for a minute. A disease that was uniformly fatal 70 years ago now has a near uniform cure rate. It's just incredible what we can do if we can bring together data for rare diseases into a single place for study. Here are the cure rates for some other pediatric cancers. And you can see that while we've done great for acute leukemias and things like Hodgkin and Wilms tumor, we have a long way to go for other tumors, especially things like solid tumors like neuroblastoma, the sarcomas, and especially AML. And when you have a child that presents with a tumor that is metastatic, the cure rates are even lower. Especially kids that have a relapse disease, we're still not very good at finding cures for those kids. So we have a long way to go with pediatric cancer, but still, if you look over the last 70 years, it's a pretty remarkable change in the status. So why have survival rates improved so dramatically for pediatric cancer? Well, the first reaction might be it's because of new drug development. Unfortunately, that's not the case. In fact, many of the drugs we use now are the same drugs we've been using for 60 or 70 years. If you read some of the early histories of treating pediatric cancer, they talk about drugs like methotrexate. I gave methotrexate last week when I was on service. So we're still using many of the same drugs, and it's been very slow to have drugs with a primary indication approved for use. How about early detection and surveillance? Of course, we talk about things for adults like colonoscopy, mammograms, but for children, it doesn't seem to make much of a difference if you detect something early versus later. And certainly, surveillance programs have not been shown to be helpful. So, what is the reason that we have had such an increase in cure rates? I think I already spoiled it for you. It's because we've been able to have cooperative group clinical trials. We've been able to bring together data from all over the country into a single place where we could study it. And of course, the US is not alone in this. There are cooperative groups all over the world that have been approaching pediatric cancer in a similar way. Having cooperative group clinical trials has led to these other downstream effects. We have a better standing of the biology and the development of molecular targeted agents. Improved risk stratification. When I was a resident, there were two types of leukemia, depending on what your white count was or whether you were a boy or girl. Now there's almost a dozen different types that you can tell at diagnosis based on the molecular characteristics of the disease. And we have much better ways to follow whether we're effectively taking care of a kid. And we have much better supportive services, much better antibiotics, much better ancillary services in the hospital. And of course, the rise of patient advocacy has brought a lot of attention to these improvements and ways to make things better. So pediatric cancer, I think, is a paradigm for thinking about rare disease, which is really a perfect storm where we have siloed data that is not interoperable. We have manual processes that are required to pull those data out. We have a pervasive lack of standards in how data are collected and shared. And we have very difficult research questions that just can't be answered with small amounts of data. A particular hospital, even for a common pediatric cancer, may just have half a dozen patients a year. How can you possibly study a disease with so little data? So it was imperative that we brought together data at scale for pediatric cancer. And I think this extends, and I hope you agree, to all rare diseases, that the keys to improving outcomes for rare disease are collecting, aggregating, and democratizing access to high-quality data. We have to get the data out into the hands of researchers. I just saw this morning, actually, and I put a slide in on it because I think it's so incredibly important. It's estimated that the economic burden of rare diseases in the US is nearly $1 trillion. And you can see some of the reasons listed here. If you think about any particular rare disease is, of course, rare. But I remember one of my mentors saying to me that all rare diseases taken together are common. And of course, we have to be thinking about the financial implications as well as the science here. So Data for the Common Good was formed to build communities, platforms, and ecosystems that can help us maximize how we use data to drive discovery and improve human health. This mission statement is purposely disease agnostic because for us, we realize that the techniques that we're using can be used across a variety of different areas. And it's not just for pediatric cancer, it can be used across many clinical specialties. And as I'm sure you'll hopefully agree, epilepsy is perfect for the same techniques that we have. Now, the tenets that we think are important are to make sure that the data are very accessible and usable. We want to make sure the data reflect our diverse population. We want to make sure that it's really easy for people to do the right thing with the data, to be responsible with it. We want to make sure that the community, that we're involving the community as we build our consensus, and that we're driving toward that impact to help improve outcomes for people with rare disease. This is Data for the Common Good. We're about 27 people now at the University of Chicago, and we're made up of programmers, communications, governance, program and project managers, and all of these folks work together to in a highly matrixed way to develop all the tools and techniques that you're going to see here today. Data for the Common Good is made up of our pediatric cancer data commons, which I'm going to spend some time discussing, our Gearbox clinical trials matching platform, which I'm not going to discuss today. And if you look at our pediatric cancer consortia, this is the way that we have it set up. Now, the way I have it here, you might think, well, we have one large pediatric cancer data commons. But the way that has actually been most effective is to have these interacting individual consortia that have their own governance over the data for which they are stakeholders. And I want you to keep this in mind, because this is going to be critically important as we talk about monogenic epilepsies and how we make sure that groups that go to such trouble to collect these data retain agency over these data. Now, in addition to the pediatric cancer data commons and our clinical trials matching platform, we've now been approached about building many other platforms in many other ways, many other disease areas. And today we'll talk about monogenic epilepsies, of course, but we're working on things like monogenic diabetes, Crohn's disease. It just came from a meeting that was studying gun violence in Chicago, looking at building a data commons for gun violence. There's just so many areas that we can look at using these same techniques. So how do we do it? You're going to see this picture a couple times today. We really believe in a big tent. Invite everyone to come together, just like this webinar. Have everyone come together to talk about the rare disease and to come up with better ways to study it. That could be academics, it could be cooperative groups, registries, patient advocates, pharmaceutical companies, whoever it is. Bring people together so that we can all work towards a common goal. One of the things we spend a lot of effort on is our data dictionary and model development. Now, this is a little bit nerdy, I know, but this is super important. People collect data in any way that they want for the most part. You could say, I'm going to have a form where I collect biologic sex as male and female, and I'm going to call it 0 and 1 or M and F. And that makes it super hard to take the data and make it interoperable. If you just take these 3 imaginary hospitals with their data collected on biologic sex in 3 different ways, it's going to be very hard to interoperate over those data. So what we do is we come up with a consensus standard and we bind that standard to a set of common data elements. Now, for cancer, we choose a standard, a set of elements that's in the National Cancer Institute's thesaurus. And so, for instance, for male, there's a code, and that code is unambiguous in its definition for what biologic sex male means. Now, you just have to have that code, and you don't care what somebody, how they spell it, or if it's upper or lowercase, because now you have an unambiguous way to collect data on biologic sex. So take that same set of data for those 3 hospitals, you can now map those to those codes, and you have a set of interoperable data. Yes, this is time consuming and expensive and a little bit painful, but as we go along, our hope is that groups that collect data will adopt the standards so that we don't have to keep transforming data from its non-interoperable form to one that we can aggregate and harmonize. This is a snapshot from one of our complicated data dictionaries. This is from our AML dictionary. And look at the bottom row here, the reasons somebody might come off of therapy. And there's a long list of reasons that somebody might come off of therapy. For instance, say the patient died or they were lost to follow-up. And so we had to work with the international community of AML experts to come up with a consensus list of reasons somebody would come off therapy. This was not easy. This took basically locking ourselves in a room together with all the experts and coming up with a consensus list. And when we did that, we came up with the acceptable values you see here. Completion of planned therapy is one of them. And yes, those are bound to those same codes. In fact, the code for completion of planned therapy actually maps to something called end of planned treatment. Those are the same thing, and now we have a code that's non-ambiguous, and we can build data collection forms that will help people collect data that is then born interoperable. This is from our website. It shows the different areas that we're working in, along with some of the metrics that we're following. So here we have our long list of pediatric cancers in blue and some of our other areas down at the bottom. We have data dictionaries established, or at least in draft form, for almost every area. We have numerous data contributors committed from all over the world. We have these consortia that we've helped form and that in many cases we help run. You can see that there's over 45,000 cases now in the data commons, with many projects in progress and many papers published. And we invite you to follow along at our website with our progress as we continue to grow and mature. All of our data dictionaries are open source. They're on our website. We invite you to download them and to use them. They're highly annotated, and we hope that people will use the data in the data dictionaries to develop their own data collection forms. A word about governance. We've been told how hard it is going to be to share data worldwide. And yes, it's hard, but when you put your mind to it, you can come up with ways to work with countries all over the world to take data and to centralize it in our data commons. We've conformed to UK data protection, GDPR, HIPAA. We have data from all over the world that we have harmonized and put into our pediatric cancer data commons. And I expect we'll do the same thing with our epilepsy data to bring the world's data into one place for study. Now, our governance is really important here. I mentioned those interacting disease consortia on the bottom left. On the right is our data for the common good team, and we act as neutral conveners. We are not allowed to give out data unless it's on behalf of one of the consortia. And then we have our international advisory groups for pediatric cancer here that help us with different areas around legal issues and ethical issues. Most recently, we started our lived experience advisory panel, which is made up of advocates, parents, and patients, which is helping us understand how we think about data sharing. I want to drive this point home. Each consortia you see there has their own memorandum of understanding. They control their own data, and we can only provide data if it's directed on behalf of a consortium. We do not give out data unless we're told to by the stakeholders. We have a cohort discovery tool that's open and available to the worldwide community. Anybody can log on and use it. This is looking at some of our AML information here. We have our Interact AML group with 4,000 patients. In the middle are the different data contributors. These are cooperative groups from around the world that have collected data. And on the right is a snapshot of the clinical trials data that make up our AML data commons. You can go in and do a faceted search using any of the terms in the dictionary, and here's a search looking at stage 1, 2, and 3 neuroblastoma. You can save that search and you can come back to it later, or you can share it with somebody else. And you can also do in-browser analytics. You can generate survival curves to try to do hypothesis generation. And so this is now a really commonly used tool for people to see, are the data available for me to study, and what are potential hypotheses for me? This is looking at our impact. We have over 90 publications that we're tracking, and impact through publications is one way to look at it, but the real impact is, are we changing the science and how we take care of kids with cancer? All right, I want to switch over to talk about what we're thinking about for monogenic epilepsy. This is a slide that Dr. Nordli gave to me last night, and it shows the development of new medications, new drugs to treat seizures. And you can see that there's lots and lots of drugs developed. Before I mentioned that there were very few drugs for pediatric cancer, well, you can see that there's a lot of drugs that have been developed and approved for the treatment of epilepsy. So have we seen great increases in our ability to treat seizures? Well, not so far. This is the data that Dr. Nordli shared with me, that over the past 20 years, we have not seen a huge progress in how we take care of kids with epilepsy and control their seizures. And to me, this is a perfect storm. We have a rare disease, we have siloed data, we have a lack of standards and interoperability, and we have really tough research questions. So to me, this is the perfect kind of area that we should be looking at to apply the same principles as we've done for pediatric cancer. Bring the data together so we can study it. Here's our tent again. When we think about building a monogenic epilepsy data commons, we want to be as inclusive as possible. So normally we would go to academic medical centers, which we are, but we're also going to patient advocacy groups. We're looking at people that have developed registries. And yes, we should be talking to industry about data that they've collected and how they are thinking about rare diseases like monogenic epilepsy. The tent has to be big. In addition to those groups, we should look at closed and open clinical trials. We should be looking at, as I said, commercial partners. And I really think it's important that we do this worldwide. There is a real tendency to want to do things like this in a very local or hyper-local way. Often we're approached with somebody that wants to do something just at a single institution. I don't think that's the right way to do this. I think we need to be thinking worldwide right from the start, because we know we can solve the problems with data sharing. And by having a worldwide representation, we're going to get such a better set of data that not only is much more diverse, but really lets us get at the super rare subtypes of disease. So as we thought about working on this, we were introduced to Citizen Health, and they become our first partner. I'm sure many of you know and have worked with Citizen Health. It was co-founded by rare disease parent Nasha Fitter. And Citizen is dedicated to empowering patients to drive their care and research forward using their own data. And Citizen wants to help accelerate the development of treatments by using research-grade data, by helping shorten how long it takes, and by driving down the cost for drug development. Citizen says they have over 80 groups that they're working with. Here's many of them listed here. And for us, Citizen is such a good first partner because it allows us getting our foot in the door for working with a lot of these groups, and allows us to think about how we can work with a partner that's been doing this for a while, like Citizen, and how we can bring our own unique processes to the table. The Citizen is using many different types of data, and I'm going to talk about multimodal data several times. They're using things like genetic data, electronic health record data, imaging, patient-provided surveys, data from insurance companies, and they're feeding that downstream, not just to researchers, but to clinical care teams, to patient advocacy groups, and to third parties like pharmaceutical companies to help drive drug development. So for us, the way that we've been thinking about working with Citizen is to tackle this area on the right of providing data for research institutions. So we have already executed a letter of intent with Citizen that we are going to be taking data from Citizen, and we are going to be providing that to researchers on behalf of Citizen. Now, I mentioned all of these different types of multimodal data that Citizen collects. The data that we're going to be getting is mainly going to be data that is EEG data, genetic data, and the clinical data. Now, Citizen does an amazing thing. The way that they get their clinical data is they get it directly from the patient using their consent. And they will pull data from the electronic health record, and they will use a combination of automated and manual means to pull the data out, and they will structure it in a well-formed way. And they will be providing those data to us to put into our data commons. What we're going to do, and you're going to hear from Dr. Nordli, is we're going to be extracting digital biomarkers from the EEG, biomarkers that can help us look at things like response to therapy or cognitive outcome prediction and other types of research. So this is a win-win situation for us because we're going to be able to take this highly structured data and to harmonize it into our epilepsy data commons, and we're going to provide the digital EEG biomarkers back to Citizen so that they can continue their important work of providing their data to other third parties to do things like drug development and to help clinicians and care teams. Now, we think about all types of multimodal data, and I keep saying multimodal because we have something really exciting to talk about in terms of multimodal data coming up. And multimodal data is not just clinical data and genetic data. We're talking about surveys and wearables and social determinants of health and other types of data. And we really want to bring lots of data together. And we want to involve a lot of groups. As I said before, not just hospitals. We want to involve patient registries, pharma companies, patients, and advocacy groups. And by involving all these groups, we're going to have access to so many different kinds of data, and we're going to be able to put those data into a common infrastructure for sharing. Now, I'm going to come back to this super important governance slide. I showed something like it before. And on the left are going to be all of the monogenic epilepsy groups that we work with, including all of these wonderful patient registries. Again, our group is on the right. We're a service provider. We're a neutral convener. We are here to help provide a way to collect and share the data. And we will definitely have international advisory groups that will help us understand best ways to use the data. And I want to drive this point home. The individual groups can maintain their independence. They shouldn't have to change their current processes. We want to help at any individual step. The goal is to help the community collect and to share data. And I hope that we have lots of questions about governance because I think it's really important that we make it clear that the Data for the Common Good team has no right to give out the data unless it's directed by the groups that put the data in the first place. So here are the different steps for building the epilepsy data commons. We're going to convene data contributors and stakeholders, and that's going on. And this webinar is certainly an important part of that process. We're going to establish official policies and procedures for governance. We're working on those. We're working on common data elements and a common data dictionary. And of course, we know that there's lots of work that's been done developing consensus standards for epilepsy, and we're going to leverage those and apply our own consensus methods to making sure that we have a data dictionary that can be used internationally with data from all over the world. We will clean and standardize and process all types of data, including EEG and clinical data. We're going to create a public search portal into which we can put the data so that people can use it. We're going to build a visualization platform for the EEG data. And then ultimately, we want to be able to develop and deploy a whole suite of analytic tools that will leverage current and exciting forms of artificial intelligence and machine learning to help us better leverage EEG. So looking at it this way, the first thing that we're doing is creating the commons, and this is what we're funded to do by Chan Zuckerberg. But then we're going to establish correlations between outcome measures for patients and the EEG data that we collect. We're going to then look at correlations between treatment and outcome measures. And then finally, we're going to pilot computer adaptive testing to look as a potential gauge for a child's cognitive function. Ultimately, we're going to take all of these data, these multimodal data that are harmonized, and we're going to use those hopefully to predict cognitive outcomes. That's one of our initial aims. But also look at things like understanding the disease biology better, develop new therapies, helping understand predicting response to therapy, and certainly looking at things like risk stratification as patients present and are treated. So I think that collecting the data is just the start of it, and understanding the important scientific questions we can answer with the data is the next crucial thing that we're hoping to define. I want to spend a few minutes talking about patient-directed data curation. There are many ways to get data out of an electronic health record, and I pulled a lot of slides up because normally I walk through these very slowly. We just don't have time today. On the left is bulk transfer of data, and this is something that we're getting quite good at, but it requires a lot of regulatory maneuvering at each institution. In the middle is what Citizen is doing, which is a manual request. The data are of such high, wonderful quality, but for us, it's not scalable to be looking at thousands or tens of thousands of patients. What we're really excited about is using something called individual API transfer, which is going right to the patient so that we can rapidly get access to data with their consent. Now, what is a patient access API or an application programming interface? That is the way that when you log onto your phone, you get data from Epic or Cerner or whatever your electronic health record is, because you have an app on your phone that leverages that API. And if we get the patient's consent, then we can do that on behalf of the patient, and we can leverage that rich data in the electronic health record for use for our downstream data commons applications. I tried this on my phone. You can launch your, if you have an iPhone, you can launch your mHealth application, and you can do this. There's going to be more and more emerging tools that will let you leverage data from your electronic health provider using an API. So what we're building, we actually have a funded project on this already, is we're building software that's going to be a front end for patients to agree to let us access their data, and we'll be able to pull data out where the patient has control for what kind of data and for how long we have access to it, and we can put those data into our storage infrastructure. The way that this will look then is the patients will give us consent. The data will be pulled out of the electronic health record into a database. Structured data we're really good at, like drugs and labs. We'll be able to pull those into our data commons after we harmonize it and de-identify it. And then the free text notes, which is like the really good stuff, is we will have a whole pipeline where we can use something called natural language processing, which is using artificial intelligence to pull out the meaning from these notes, process it, and add that into our data commons. We can't ignore the free text notes just because they're difficult, because that's where some of the most exciting information is on patients. So I think the future of data collection looks like this. You have patients that through an interface will control their own data, and that's data from multiple institutions. And I think this has been such a vexing problem in trying to understand how can we combine data from multiple hospitals. The patients can do that through a patient-facing app. They can decide which hospitals, which data. Those data, once they consent, can be put into secure storage and processed. And then we can use that data for all sorts of downstream applications. People can write apps against the data. They can go into a registry, and they can end up in our data commons. This is looking at the different types of data on the left, the medical records like patient provided, I just talked through those, but also surveys, patient-reported outcomes, text messaging, micro-engagement, and then the standard ways we think about clinical trials and other types of records can all go into a registry, be de-identified, and end up in our research data commons, where we hope to put tens of thousands of patients' data. I want to talk about one more thing. I'm bumping up against my time, but we're just so excited about this, is looking at how we look at multimodal data and artificial intelligence from an ethical perspective. So I talked about all these types of data that are going into our data commons. We call this multimodal data, so many different modes of data. EEG data, genomic data, clinical data, wearable data. And we're going to use those data for all sorts of things like predicting outcomes and predicting response to therapy. But using those data comes with a lot of responsibility. So artificial intelligence and applications that rely on these multimodal data are growing so fast. And you know, just from using ChatGPT and other types of tools, that these things are coming out every single day. But a number of limitations constrain their use, and they should constrain their use. Issues around trust and transparency and fairness and equity. And by incorporating an ethical framework and community engagement throughout the entire life cycle, we can try to implement these tools more equitably across the community. So again, looking at these types of data, we want to think about things like, what are the rights of the vulnerable populations that we're studying? What is the risk of re-identification for these very rare diseases? What about consent for patients or assent of young adults? What about reconsenting patients when they turn 18? What about the dynamic between parents and children when it comes to using their data? We have to be thinking about all these issues and many, many more. So we were very fortunate and just found out yesterday that we have another initiative funded by Chan Zuckerberg, which is to look at multimodal AI ethics when it comes to our epilepsy research. So we're going to be co-creating a framework for the ethical implementation of multimodal data. We're going to be partnering with leaders in biomedical ethics to co-develop this framework for developing these AI models. We're establishing a stakeholder expert panel, which I'll show you in a minute, to give us feedback and to consult with on how we do this. And we're going to put that new framework into practice by creating these artificial intelligence models that incorporate these really exciting multimodal data. The name for this project is a little cumbersome. It's Community-Oriented Design and Ethics for Pediatric AI Resources and Monogenic Epilepsies, or CODE-PAIR-ME. You have to have a really good acronym for your projects. This is our epilepsy stakeholder panel. You'll recognize many of the names here, including the top name, who we're going to hear from next, Justin. But many of the other folks here, I'm sure, if you're in the epilepsy community, are going to be names that are well known to you as stakeholders. We're going to be working with the stakeholder panel to design our ethical framework, things like identifying our ethical concerns, engaging stakeholders, translating the stakeholder values into constraints and metrics and how we can measure ourselves, and then providing accountability on what we do back to the stakeholders. So we want all of you to think about getting involved. We want you to think about becoming a member of the Commons, to contribute what you know, to help us with our registries. It doesn't matter from where you come. We want you to be involved and get under the tent. We want you to participate in the Commons development through stakeholder groups. We want to have you consider being on some of our expert panels, and we want to have you advise us and contribute to how we develop our standards. You can always contact me. You can contact Bailey, whose email address is there. There's so many ways to get a hold of us. So let us know if you want to be involved, because we'd love to have you with us. So I think what makes to bring it back, I think what makes our group pretty unique in this space is that we're really all about the data. We work on the data model and on the structure of the data. There's a lot of groups out there that will take data into a data lake, or they'll make up a standard and just shove it in there. That's not what we do. We have a lot of respect for the data model and for the standards, and we won't take any data in until we have a stable consensus dictionary. We want to make sure that the dictionaries that we build are not only harmonizable across the things that we see with many diseases, but also are deep and meet the highly specific needs of that particular clinical area. We are really intent on having the data centralized and harmonized in a single place. We think that's really important for study, but we want to make sure that the groups that contribute the data retain autonomy and retain their own ability to control how the data are used. Change management and quality control are hugely important. Have the data changed, has the model changed, have the tools changed. This has to be tracked and has to be apparent to anybody who's using the data or the tools. And as I've talked about many, many times, the governance is critical. We have to make sure that people understand how their data are being used and that the folks that work so hard to collect the data retain control and are able to understand exactly what the responsibilities are of every group that's using the data. Again, this is our group. We continue to grow. We'd love to hear from you. We'd love to have you get involved in this and any of our other projects. We've been really fortunate to get a lot of funding from foundations, and I talked about our Chan Zuckerberg funding today, but we have had such great benefit from all these foundations that have been able to give us funding to start these different initiatives. Of course, we've had some National Cancer Institute funding, we've had some government funding, but really it's the foundations that take the gamble of funding infrastructure, which is so critically important. And so we're just so grateful to the foundation's support, many of those foundations that you're part of. And of course, extremely grateful to our patients and families that make our work possible. And with that, I'm going to pause briefly. I'm actually over time. So what I think I'm going to do is I think I'm going to go ahead and move on to our next speaker. I haven't been able to look yet, but I see a little thing that says there's like 21 different comments. So I assume there's been lots of great questions, and I'm going to work hard to try to answer some of those in the chat, but hopefully we'll reserve many of those for our panel discussion coming up. So it's with really great pleasure that I introduce Justin West. Justin runs the KCNT1 Epilepsy Foundation. He's going to talk about his experience with his son, Andrew. He is a physician himself, so he brings a particularly unique perspective. He works with researchers and industry to identify and evaluate different therapeutics. And I got to see Justin's slides. They're very exciting, and I'm really looking forward to hearing him speak. And then I'll be back to bring us through the rest of the webinar. So I'm going to stop my share. And I'm going to invite Justin to share his slides and turn his mic on, which it already is, and to go ahead. So Justin, it's all yours.
All right. Let's see. This out of the way, and I will try to move this bar so I can launch. Here we go. So thank you very much. Can everybody see the slides and hear me okay?
Perfect. Looks good.
Okay, great. So thanks very much for having me here. My connection, I'm both a rare disease patient myself and the parent, obviously, of a rare disease child. My connection here is through Doug Nordli. I had the fortune of having Doug during his very brief time at Children's Hospital Los Angeles, help diagnose Andrew and take care of him. And we feel really grateful that he was part of our journey. He continues to be associated with us as a key member of our science advisory board. We started talking about this program for the Data for the Common Good a couple years ago, and it's really exciting to sort of see this come to fruition and to see the potential impact it could have for the epilepsy community. So my son has KCNT1 related epilepsy. It's a de novo autosomal dominant gain of function mutation. There's two main phenotypes. There's an encephalopathic epilepsy, which is massively drug resistant. Our children have hundreds of seizures daily, and there's total developmental arrest. There's also a less severe phenotype that mostly involves nocturnal seizures and with less compromised development. So to get personal quickly, this is me with Andrew, day 0 of life. I live in the OR. This is a pretty natural environment to me, but what was not natural to me at all was epilepsy. So Andrew was born in April, our third child. He was totally normal, hitting all these little mini milestones until 4 months, and he developed this little flickering of his foot. We were told it would be fine. This is before we saw Dr. Nordli, that it wasn't going to be a big deal. He might have to have an epilepsy surgery one day, but he would be a very normal child. Eventually, we got gene panel testing. We had a massive delay in diagnosis, which I think a lot of us in the epilepsy community suffer from. And I'll never forget Doug calling me. I was in the PACU after my last surgery of the day, and Doug called to share the diagnosis, and it really changed our lives forever. This is a destructive diagnosis. For our patients, they have nothing resembling a real life. We have parents who lose jobs, get divorced, live with anxiety and depression. Most of them are medicated. The siblings become caregivers. They suffer through the loss of a sibling. They're ignored by parents who are focused on their sick child. They're teased by children who don't understand wheelchairs. They go through their own anxiety, and most of them are in therapy. Even our grandparents, they become caregivers all over again, helping to care for their kids who are caring for sick kids. Our families are just exhausted. For us, it means spoon feeding every meal and squeezing every cc of water into Andrew's mouth. It's changing every diaper and transferring this ever-growing child with an ever-aging back. It's trips to the emergency room. It's weeks and months in the hospital. It's fighting insurance companies who want to deny us life-saving equipment. It's fighting a school system who has no idea and doesn't understand to this day why he's not there. And it's working full-time jobs while caring for not just sick kids, but for their siblings, and ultimately not sleeping, which many countries consider a form of torture, and we're all living with that on a daily basis. In terms of what to do with this disease, one of our doctors, because when you have a rare disease, obviously you tend to see many experts to try to find the one who has the right answer for you. But the one that stood out in some ways was the one who said, there's no treatment, there's no drugs in development, take him home and wait. And that didn't mean wait for something good to happen. Fortunately, Andrew had other plans. We were told he'd never learn to sit, and he learned to sit. And then we were told he wouldn't really use his hands, and he learned to use a bottle. And then eventually he learned to stand, albeit with a fair amount of assistance. I love that photo on the right because it's the one that makes him look most like my other children. Andrew has the DE phenotype. He's had well over 50,000 seizures. I went through life, medical school, residency without ever seeing a seizure other than what I saw on TV. And to witness 50,000 plus seizures it's deeply impactful. Andrew is on any given time on 5 different anti-epilepsy drugs. He's now 7 years old. He has still never said his first word. He's never taken his first steps, and he depends on caregivers 24/7. If it wasn't, my wife's also a physician. If it wasn't for heroic efforts on her part, running a mini ICU in our house, Andrew would not be alive. For our patients, time is really critical. We lose half of our patients before the age of 10. Recruiting families to studies, if we ever get to there, is really challenging because they've been destroyed by this disease. And recruiting those families for trials who have transitioned from fighting to acceptance is going to be a real challenge for us. We do not have time to wait for a perfect drug. Our children are at risk on a daily basis. And on a monthly basis, we get photos sent to us like this when we lose another one of our warriors. The way I think we look at it is we need a good drug now, a better drug in 5 years, and a cure in 10 years. On this call today, we've got patients and families, researchers, clinicians, pharma, and FDA members. For the patients and families, I would say, from my perspective, the math problem is pretty simple. You can invest all your time and resources into trying to develop a drug knowing you have a 90% chance of failure. Or you can create a community garden with all of the data and assets that you need for pharma to come in and plant the seeds that might turn into life-saving therapies. I would highly encourage anybody here to share your data. I think that HIPAA is a really important thing. We talk about it in medicine all the time, all the ramifications. In my opinion, it's been a bit detrimental to research efforts because we have this cultural shift to protect data. Whereas when you're a rare patient, the best thing you can do is share your information with as many teams as possible. So trying to fight against that, I think, is important. The things you can do: sign up for your contact databases, sign up for the registries, participate in the natural history studies. And when you're asked to volunteer to provide blood, skin, and any other biospecimens, just do it. Because everyone in the food chain you need to help you go from a diagnosis to a treatment needs that data to work with. It's really critical. For the researchers, I think that something that we can communicate is that we're all grateful to anybody who studies our rare diseases. My plea for the researchers would be to pivot away from traditional siloing of data. Our community happens to be pretty lucky. We have researchers who have been really open-minded to sharing data and insights so that we can all, as a community, advance the understanding of our disease. But we do have teams that are still stuck in old traditions. We have one group in particular that's been sitting on actionable data for a repurposed drug program. They've been sitting out for 2 years waiting to publish. So I take every opportunity I can to remind them that we lose a child every month, which means they owe us 24 lives. So don't hold on to your data. For the clinicians, I think it's really important. Talk to your patients about how important data sharing is. Most of our parents that we talk to in our community have never had that conversation with their physicians. It's really helpful if you could encourage them to sign up for the registries and the natural history studies. I think the foundations get good at networking with our parents to encourage them, but the clinicians have that caregiver relationship and the trust that comes with it if you've been providing outstanding care longitudinally over time. So take advantage of that opportunity. Your efforts are really critical to what we're trying to do. For pharma, I guess my ask would be to come to Foundation sooner. Don't come to us 5 years into a program when your preclinical package looks good. It doesn't give us much time to gather the data that you eventually tell us that you need. I think really we should be collecting data in parallel with your early discovery program so that the natural history studies and biomarkers and other critical assets are ready to go when you finish NHP talks. Working in series for rare disease communities with significant morbidity and mortality ultimately costs lives. For the regulatory folks, we recognize the substantial burden that you have on ensuring the safety of Americans. The flip side of that, though, is the burden on parents and patients that is incurred while waiting for new drugs. As of right now, my son Andrew is on 5 different anti-seizure drugs. None of these drugs target the mechanism of the disease. None of these were ever tested on KCNT1, and certainly none of them were ever tested in the crazy combinations in which we give those drugs. Over 7 years, we've tried combinations of 15 different drugs, and unfortunately, this isn't unique to my son. Our average patient tries 13 different drugs over a lifetime, and we know through Citizen, because we participate, and we're very grateful to, that 90% of our patients are on 5 or more drugs concurrently, even though we know that goes right against what we consider to be the standard of care. Our community doesn't really need the best drug in 10 years. We need a good drug now. My son can either die from complications of his KCNT1 diagnosis, or he can die trying a drug that might bring back some part of him that we've lost. I know the risk my family is willing to take, and I know the risk that our community is willing to take because we have the data and we're willing to share it. Thank you.
Thanks, Justin. That was really great. Stay on for 1 sec. We're going to break in a sec. We have a 5-minute break, but I just wanted to, and we'll have more time to talk during the panel because we did get a lot of questions for you. But I just had one question right now. Obviously, as a physician, you had a unique perspective with your son, but most people who have children with rare disease are not physicians. And I'm curious, how are we failing patients who go on this journey, and what could we be doing as a community to help people wind their way through the system a little bit better? Because it's just so frustrating how long it takes to get diagnosed. It's often a who you know kind of thing. So I'm just interested in your perspective on that.
I think I could create a laundry list for you of ways in which people fail. We're a 2-surgeon family and we struggle with Andrew. I think that, one of the points is paternalism is still a massive problem. I think that we have clinicians who still think they know, what's best for our families. When you have a disease that is, ultra rare, you can't possibly know much about it. They don't really rely on us for information. They don't really do a great job always laying out the various options that we have. And so they take it upon themselves to make decisions for us without giving really truly informed consent and really giving us the perspective of what the disease is going to look like over a lifetime. So I think that, better education on a clinician's point of view. I mean, I say that as a clinician, knowing that one of the many reasons that we're stuck in this position is time, right? So reimbursement to physicians goes down all the time. The cost of seeing patients goes up all the time. And our systems aren't really well set up for patients and clinicians to have long conversations. And so I'm not putting the blame squarely on the physician, but the system is not perfect. I think that, it's also a failure. When you have a rare disease, I think that part of it is, parents like us end up learning about our disease through Google. And it's not really how we should be doing it. In an ideal world, you not only would you get that piece of paper from Invitae with your genetic diagnosis, but you'd be handed a playbook that says this is what the next 18 years of a, your child's life looks like. It's hard to do that for every disease, but most foundations like ours are making efforts to do that. But with the genetic epilepsies, there's a lot of common denominators, and I think that putting together, resources and making those resources infinitely available would be very helpful, so that parents walk out with that devastating diagnosis, but have some semblance of a plan and hope. We were also never really taught by our physicians, how do we go about, if you don't have a drug in hand, how do you go about advocating for one? How do you start a foundation, and how do you plant the seeds to contribute to research, basic science, and translational programs so that you can go from not having a therapy to having a therapy? I mean, the sad truth, 5% of rare diseases have a therapeutic. And if there was better, if we had, I think, a better culture that encouraged parents recognizing that many of these people are intelligent, talented, and are infinitely motivated to save a life, many of us have the skill sets or can learn the skill sets to go and advocate aggressively and successfully for our kids, but we end up doing it ourselves, and we learn to do it from our fellow parents who are struggling. We don't learn it from the system, and I think that's a shortcoming.
So maybe creating more contact points, resources, navigators. I mean, I think there's probably a lot of solutions. The question is, what's going to be something that's sustainable and scalable and something that can be funded, right?
Yep.
Okay, so I think we're going to, Justin, we have a lot of other questions for you, and I think I'd actually rather ask them in the panel because I want to also be able to ask Larry and Doug during the panel. So if it's okay with you, we'll switch over to the next speaker now, and I think we're just going to go ahead. We're actually going to skip our break because we were getting tons of questions, and I want to make sure that we can get to everything. And I talked to Dr. Nordli, and he is indeed available. So I'm going to share my screen again. And I'm just going to introduce Dr. Nordli. So Dr. Nordli, I've known for a couple of years. Doug is section chief of neurology at pediatrics here at University of Chicago. He's an epileptologist and neurologist. He focuses on pediatric epilepsies, and he's particularly focused on early onset epilepsy. And I wouldn't be surprised if almost everybody here knows Doug, because every time I go somewhere with Doug, we meet people that Doug has either taken care of or he's taught. So it's been really humbling to see the impact that he's had. He's looking at techniques to predict the course of epilepsy and how we can find out better ways to predict outcomes. He's the co-investigator with me on our Chan Zuckerberg grant to build a monogenic epilepsy data commons, and I'm super excited to invite Dr. Nordli to speak. I'm going to turn it over to you. So you should be able to share. Make sure you come off mute, and I'm looking forward to your talk.
Thank you so much, Sam. And I'm the Felix Unger of neurology, and always seem to be suffering from seasonal allergies. But also, that was a very powerful talk from Justin. Can everybody see my slides? We good?
You got to go into presentation mode, and
okay,
perfect.
So I thought I would start off just by way of explanation of how I came to this collaboration with Sam. And one of the ways that I thought would be maybe useful is to talk about this second opinion clinic. So a few years after getting to University of Chicago, I decided to form a second opinion clinic, which was purely consultative. And over the last, I'd say 3 or 4 years, I've seen over 250 very refractory patients. And the way that it works is that people send me data. And so I go through their data. We actually look at their, EEGs from their CDs, study their MRI scans, look at the genetic data. And then Audrey Otomo, who works with me as a nurse practitioner, helps me in aggregating that, but also in talking with the parents. And then she and I go on and spend more time talking with parents, and mostly that's focused on the semiology, but also other factors that about the illness. And the primary thing that I've learned about this, the critical thing, is that the reason many times that somebody doesn't have the right diagnosis is that people have been too pressed for time. They haven't had the time to sit and actually look at the primary data. And of all of the primary data, I found the EEG to be consistently the most helpful. So here's an example of a patient. This is the patient that I saw at 8 years. The absence had started several years before this, and it proved to be refractory to very well-chosen medications for absence epilepsy. So those of you that maybe on the webinar that might be EEGers will notice that there's something peculiar about this EEG. There's two things that I would point out is that the background is slowed and doesn't have the normal organization and complexity. And then, of course, there's too much slowing, and there's a particular type of slowing that's going on. There's a rhythmic delta activity that's frontally maximal. And you're seeing these spikes that are admixed. And you might say, well, kind of looks like childhood absence epilepsy. But then as you look at it more, you say, no, this is not typical for CAE. And then you could say, well, maybe it's a little bit of an older patient, so it's more like JAE, but still really not looking consistent with that. So thinking that, this is not just a genetic generalized epilepsy, but a mimicker of that, and seeing this pattern, pretty quickly I had a thought as to what this might be. And that was GLUT1 deficiency state. Now, why did that come to my mind? Because I trained in New York with Daryl DeVivo, who discovered GLUT1. And one of my tasks as his assistant was to go through all of the EEGs on the first cohort of patients that were identified in the world. It was, at that time, it was about only 25, 50 patients. So we reported on these findings and look at this EEG. Like, I'm sorry, it's poorly reproduced here. It's from the PDF of the original publication, but right, it looks pretty much identical to the EEGs that I just showed you. So on the basis of that, we did genetic testing and saw that, yep, indeed, the patient does have GLUT1 deficiency state. But we knew a lot more than that. Because of that, and because we had secured the diagnosis, we knew how it impacted the brain. And this is an article that Juan Pascual published along with Dr. DeVivo and others, showing that there's a particular region of the brain that's affected with GLUT1 deficiency state, and that's the thalamus, which makes sense, right? That's why you get these thalamocortical projections that are so aberrant. So we knew what kind of epilepsies that you see, what the EEG signature is, and we also, it came to our minds at the time that we had a potential treatment. So here's the logic behind it, that in this case, you have glucose that comes in and normally is a preferred substrate that our brains like to use. But if you can't get glucose because it can't cross the blood-brain barrier and get into the CNS, then over time, your brain gets depleted of these resources, and it's kind of starved of its basic nutrients in order to power activity in the brain. What you can do is you can put people on the ketogenic diet, increase their ketone bodies, and shown here is beta-hydroxybutyrate, and through acetoacetate and acetoacetyl-CoA ultimately gets converted to acetyl-CoA and comes into the Krebs cycle or the TCA cycle. And this is the very powerful portion that produces a lot of energy and rescues almost completely the phenotype, but not completely. So from atypical response to drugs, EEG signature, genetic diagnosis, we get a fairly precise treatment that, as Jörg Klepper showed in this article, works pretty well. But if you look at his concluding sentence in his abstract, we still need to provide new opportunities to improve treatment in the condition. But we came a long way in that process. Now, let me show you, in contrast, another patient that I was asked to consult on. This is a 2-year-old who's having drop attacks. And on the left-hand side, what you're seeing here is a big burst of spike. It's a very high amplitude, diffusely distributed spike. And then afterwards, this relative attenuation of the background that's pretty profound. And this is the time period where the patient loses stasis or an astatic or an atonic seizure. And at first, I thought, oh, maybe this is Doza syndrome, myoclonic astatic epilepsy. But as I looked at it more and how chaotic, which sometimes the EEG can look pretty chaotic, I noticed that the background was consistently slowed. And this was even more ominous, that there were these epochs where the background completely attenuated, which is not at all typical. So then come to find out through genetic testing that the patient has a mutation, but it's a very rare mutation and very little is known about it. So you go into the literature, look at what is the phenotypic expression of this mutation, and it's mostly in genetic journals. So you get some description of the seizures, but it's not really the kind of description that an epileptologist would give you. It's useful, but not terribly detailed. And you get even less information about the EEG characteristics. So now you're in a situation where you actually don't have an electroclinical syndrome diagnosis, except, DEE. Really don't even know for sure if the gene is causing this problem because it's not a clear pathogenic mutation. And most importantly, have no clue as to what is going to be the ideal precise treatment for this patient. Yes, we can manage, of course, based on the seizure type, as we often do, but it's a far cry from the first patient that I discussed. And so the reason I'm going into this and painting this picture is that that's why I was so excited to meet Sam several years ago when I arrived here at University of Chicago, because I've long thought for more than 30 years that what we really need to do in pediatric epilepsy is the same thing that pediatric oncologists did for cancer. And the key to that is figuring out ways to have data repositories where information could be shared, just as Sam said. So we've talked about this for several years, and then finally this opportunity came up, and so we're really excited about this opportunity. Now, in the chat I noticed a couple of questions, and I'm sorry if I'm not picking up the chat right now. Maybe I should pay attention and do that. But one of the really good questions is, why monogenic epilepsy? So let me try to explain this. So first, like Sam was saying, okay, each monogenic epilepsy is relatively rare, but in aggregate, it accounts for at least 30%, and I would say probably upwards of 80% based on our EEG data of epilepsies that fit into this category of refractory epilepsies with encephalopathy, or DEE, using the more current term. So each specific gene may not have as many patients and constitutes true rare diseases, but in aggregate, this is a really important topic. And each one's pretty precise, right? So you can look and identify, in many cases, a pathogenic mutation and know, in many cases, for sure, that that's what you're dealing with. So a very homogeneous patient population, right? Kind of like what Sam was talking about subtypes of ALL is a direct correlate, and the ability to prognosticate and select treatments based upon that. And we also thought that this is an important area because then, as we learn more information about what the gene is doing, the opportunity for other precision treatments comes up. So up at the top is what we're doing right now, right? Our conventional epilepsy treatment. We gather some phenotypic characteristics, we counsel, and we try a treatment, but more than half the time it's not ideal and it doesn't work well at all. So here's our promise, is what has made this era for all of us right now so exciting, is that yes, we can still collect these phenotypic characteristics, and it's still very important to listen to people and hear the details about their illness. But now, through the gene, we can study the gene and the impact that the gene has, whether it's in vitro or in vivo, and we can devise any one of a number of different potential treatments. So it could be that we use existing drugs, just like Sam pointed out they did with pediatric cancer, that we just haven't figured out how to use existing drugs correctly. Or we could repurpose drugs, or maybe there are small molecules. I heard a really nice talk from Rima Nabu last year from Enfant Malade, and she said we really shouldn't stop thinking about small molecules. Or maybe treatment B is going to be an ASO, right? Or maybe treatment C would be actually an AVI vector and actually insert a corrected gene. But that's why our attention was drawn to monogenic epilepsy, because in aggregate, they're critically important, and they offer this opportunity to kind of push us forward, hopefully more quickly in terms of discovery of cures in pediatric epilepsy. Now, Tracy Dixon Salazar raised a really good point, is should it only be monogenic epilepsies? And in answer to Tracy's question, I think, no, it should not. And let me touch on that a little bit in the future, in a few more slides. But right now, this was our grant proposal to CZI. This is what we intend to do and accomplish for satisfying that grant. But I think it is very logical to broaden it, as Tracy raised. So you just heard from Justin, and I've been to 2 CZI meetings, and it's simultaneously refreshing and inspiring seeing what families are doing, and it's exhausting just hearing what they've been through to get through this journey. They establish foundations, they have to gain members, do website development, educate, as Justin said, fundraise, hire staff, engage key opinion leaders, engage scientists, establish registries, do advocacy, all the things that he mentioned, even before you get to preclinical research. And then you have to determine what are going to be the patient-reported outcomes. You have to join or create biorepositories such as Combined Brain, or you have to start or join a data commons. And multiple examples, maybe multiple of these groups are on, including Citizen Other, and then engage in natural history studies, which Citizen just reported is one of the aims that they are going to be focusing on. You have to engage the FDA, and we'll be hearing from the FDA a little bit later, and pharma, develop animal models, and find biomarkers. And I want to focus on this. But then you have to do all those things that I just mentioned in terms of study treatments, in the hope of ultimately conducting a clinical trial. And I think I cannot improve any possibly on Justin's comments about that, to find a cure. And then after that, you need to probably do some kind of post-treatment surveillance. So it's exhausting. I don't know how people have done it, but we need to help. There needs to be a more streamlined roadmap and help from multiple different facets to help these foundations get through all of this. But I want to just focus for right now on finding biomarkers. So I mentioned that EEG is really important to me, that I use it all the time in my clinical practice. I still think it's one of the most invaluable tests. It's a very sensitive test of ongoing brain function. I'm on inpatient service right now. There was a patient that was on service for a different problem, but the ascertainment from the team was that the patient was doing well, but the EEG showed otherwise. It's so sensitive. It's so sensitive to brain function, but it's etiologically nonspecific, or so we think. It's universally digital, so we can easily store it and share it, and that allows quantitative analysis. And the findings can easily be abstracted and categorized. I want to just share my thoughts on that with you. So let me show you this EEG. So there's multiple ways that you could approach this, right? You could say, well, it's a non-specific EEG, it's abnormal, there's epileptiform activity, there's changes in the posterior rhythm, there's admixture of slowing, there's bursts of rhythmic frontally dominant delta activity. You could do all that, and you could actually probably enumerate dozens of features about this EEG, or you could abstract it in some way. And you could take those features that I described, slowing, background disorganization, multifocal and diffusely distributed spikes, and you could lump it into one category. Now, this EEG you can even subcategorize, and many of you out there, again, if EEGers would recognize this and would know pretty quickly, like that one of the things that needs to be considered And the differential diagnosis because of this notch delta is Angelman syndrome. But if you didn't recognize that, you'd at least know that it belongs in this category. So this is what I mean by abstraction of the EEG. We've done this for over a decade now in multiple places. I've taught a lot of people this, and it seems to be helpful that you can break up EEGs and epilepsy into 5 different categories. And a big discriminator is whether the EEG background is normal or abnormal. And so in the northern hemisphere, the background's normal. In the southern hemisphere, the background is slowed. In the western hemisphere, the findings are diffuse or multifocal. And in the eastern hemisphere, they're focal findings. And so you can kind of very simply, based on that abstraction of the EEG, segregate into these buckets. And then within these buckets, based on the age of presentation, you can usually narrow it down to 2, possibly 3 in some cases, electroclinical syndromes. And many times the prototypic seizures, the seizure semiology, combined with that gives you the answer of what specific electroclinical syndrome it is. So here's just an example of how you can do that over age, these different categories, and you can put the EEG syndromes in there. So there is something else that we need to do, though. We need to take these epilepsies with encephalopathy and we need to subcategorize them further. And I think this is entirely possible. Remember that EEG I showed you with that rhythmic frontal delta that's notched? It's not only in Angelman's, as many EEGers know. You can see it in other conditions as well, including copy number variants. So it's a pattern, right? Just like there are dosimeters and there's Jevons mimickers and so forth, you can subcategorize these epilepsies with encephalopathy into their characteristic EEG pictures. So why EEG? First of all, it confirms epilepsy. Second, you can categorize the findings, as I said, and validate the epilepsy syndrome. So getting to Tracy's point, in the course of doing this, you can arrive at that. And so say that's infantile spasms or Lennox-Gastaut syndrome. We could arrive at that and validate it with the EEG. And that could be a very important other way to approach this data set. And then you can try to subcategorize further that might give you clues to the cause and the pathogenesis. And all of this is important, but the big additional thing that I think EEG can do for us is it can serve as a biomarker for progression or hopefully remission of disease. So 2 critical things that I think about when I'm caring for a patient with a monogenic epilepsy is seizure control and normalization of brain function. And seizure control, we can use seizure diaries. Parents may not detect every seizure, but in my experience, they're awfully darn good and pretty darn consistent in detecting seizures. So I don't think that's as big of an issue, although there are exceptions. But I think what's harder to get at is normalization of brain function. You can rely on patient-reported outcomes. Those are really critical. But industry and others are probably going to want additional things. But what is that? A Vineland, that could take months to change, right? Maybe even longer. Neuropsych testing, perhaps even longer, right? Even after successful epilepsy surgery, it may take up to 5 years to see improvement in neuropsych testing. And it's subject to test-retest issues. And then comes the EEG. So you might say, like, Doug, why do you think that could be a helpful biomarker? Because for almost now 100 years, it's known that with cognitive changes, your EEG changes. The first report of that was from Hans Berger himself. More modern, I really like this. This is really nice publication from Boston Children's. And here is a study that looked at patients with Dravet syndrome who had studies that were obtained in sleep. And so different from what I would normally look at awake alert background, but this was quite clever, looked at sleep. And so you can see differences in power here. Let's zoom in and look at this. So these are patients with Dravet in red and control over here. And this is a frequency. It's on the abscissa and relative power over here on the ordinate. This difference in delta power shown here, controls in Dravet and sleep, and theta power was highly predictive. And you can see that as opposed to the normal acquisition of increasing power in these frequency domains, it remains relatively stagnant in the patients with Dravet. This is just one example. There are many other examples that you can find that show that EEG may be a very useful biomarker in the monogenic epilepsies. But it's not going to be one size fits all, would be my prediction. So up here on the left, I showed you this EEG comes from a patient with Angelman syndrome. Down here, as Justin was speaking about, is an EEG sample of someone with KCNT1. Up on the right, over in here, this is SYNGAP. And you see this Jevons-like appearance, slow eye closure, ramp up of fast activity posteriorly, and diffuse spike wave discharges. And finally, over here is Dravet. You don't need to be an EEGer, right, to look at these 4 EEGs and say these are very different. So the quantitative measures that you're going to use to track disease is going to need to be customized to some extent. Yes, most of the time you're going to see an increase in power in the slowing in the awake tracing, but you need to be more specific in order to make more sensitive measurements. So on our team, we're really fortunate. We have a signal processing expert, Dr. Van Dragelen, and Wim has created a pipeline that is, to some extent, things that probably everybody is familiar with. From a single channel property, you can look at the time domain, and you can look at typical spectral parameters that you and I, as physicians who do epilepsy are using every day. And you can also do multi-channel properties that we also sometimes use, where you correlate different parameters. Like in the time domain, you can do cross-correlation, or in the frequency domain, you can look at coherence. And Wim has done something novel and has published on this. He's created a different way to analyze EEG based on a theorem from the 1970s of visual analysis, triple correlation, and he's applied this successfully to EEG. So the results are kind of abstract, probably, but on the right-hand side, I want to call your attention to these figures over here. This is just a normal child at 2 months old, and each one of these bars is a motif, and the motif is corresponding to the interplay in time and in space between 3 points. So you can these can translate into very straightforward things like feed forward, feedback, correlation between 2 signals, coherence. All of this is embedded. Phase coupling is embedded in this as well. So it's a way to have a snapshot of what's going on in the entirety that is unique. The triple correlation is unique and pretty comprehensive in terms of capturing that. So we just put it out there as one of a number of tools. Of course, the pipeline includes standard things that I already mentioned, but the point isn't so much as, well, is it U of C that will identify all the features? Absolutely not. Once we get the EEGs in here and this data is in the public domain, then institutions across the world that have clever ideas about how to analyze it from a quantitative standpoint will have access to that. So our job is to de-identify the EEGs and to collect mutation details, to store the digital EEGs. We're going to curate the samples so that they're very usable. We'll visually analyze by the categories that I told you, and we will also post the summary data on the website. And in addition, we'll perform these quantitative analyses through the pipeline. And overall, the purpose is to share the data. So why are we doing this here? For me, this has literally been one of the major dreams of the last 30 years for me is to have this kind of initiative going. And I'm so grateful to meet this team, Data for the Common Good, and to leverage the experience in pediatric oncology for us in pediatric epilepsy. We're lucky that we have lots of experience with EEG, and we have excellent signal processing teams here. And we're super fortunate getting the funding from CZI to initiate all of this, and we're deeply appreciative. So with that, I hope I've been judicious in terms of time. I'll I'll stop and stop sharing the slides now.
Thanks, Doug. We're going to get to Larry in just a sec, but there was one question that I wanted to ask you that came up in the chat from Heidi. And Heidi asks, ultimately, won't you need patients that have multiple EEGs over time and clinical assessments associated in time to develop a true functional biomarker? This will be a challenge for data collection. And we've talked about this, but I'd love to hear your thoughts on that for a few minutes, and then we'll move over. Thanks.
Absolutely. I couldn't agree with Heidi more. This is going to really be critical. And we've had a number of conversations as we've been discovering the landscape, talking with people in various groups, including Terry Jo and others that may be on this call, that it will be really important to match the EEG features with some measure of how the patient is doing. Multiple ways that we can look at that depending upon what kind of data that we can collect. But that is going to be a critical step. It's not hard. In my experience, we can get data, EEG data. Citizens done a really good job of acquiring that. And we can, because it's time-stamped, generally speaking, and if we know the birth date of the patient, it'll be very easy to keep it orderly in terms of what age was the patient when the EEG was acquired. So I don't think that'll be hard. It'll be really important, like Heidi's saying, to make sure ultimately in that subsequent phase that you mentioned, Sam, that we correlate some measure of clinical outcome with the EEG.
Right. Thanks, Doug. And we've had a lot of experience with pediatric cancer, longitudinal data collection as well. So we're certainly teed up to do that. Okay. Thanks, Doug. We'll have you back in a few minutes for our panel discussion. I want to go ahead and introduce our final planned speaker before our panel. Go ahead and share one slide. So probably the best thing I did in 2023 was recruit Dr. Larry Hunter to University of Chicago. He's really one of the founders of bioinformatics and biomedical informatics, publishing some of the very first papers on natural language processing and machine learning predictions for molecular function. He's also started a whole bunch of really cool conferences, one of which is in Hawaii. So he's just done so much exciting foundational work in this field over the past 30 years. His interests are very wide, and he does all sorts of cool research today, and I'm super excited to have him tell you today about his work for the Chan Zuckerberg Initiative on monogenic epilepsies. So I'm going to stop sharing and hand it over to you, Larry.
All right, hold on. I tried to share too soon. Let's see if I can do it again. Yes, there we go. Thank you, Sam. Yeah, I'm still getting a message that, let's say, replace current share. Does that work? All right, I'm going to assume that you can see my stuff here. And thank you. And this is going to be a little bit different. I'm the token computer scientist today. And I found all of the talks leading up to this both compelling and fascinating. And I want to tell you a little bit about how we're going to use AI and language modeling in this community to try and advance the progress. And Sam suggested I give you all a brief introduction to language models. And you're surrounded by them. I don't think anybody has escaped from ChatGPT, and most of you have probably tried it some. But I think in general, we lack the background to understand what it means and how to make sense of what these large language models do. And I think the best way to understand that is to think about a Jorge Luis Borges story called The Library of Babel. And if you know Borges stories, you probably know this one. And if you don't, I recommend you read it. It's a short story. And the conceit of the story is that the Library of Babel has all books of exactly 410 pages. And in it, every possible book that could be written of 410 pages. And so if you do the math, if there are about 2,500 characters on a page of a book, and there's about 100 characters in the alphabet, so 26 upper and lower case plus some punctuation and stuff like that, that leads to this absolutely unbelievably large number of 10 to the 2 millionth different possible books of exactly 410 pages. So, and the Borges story is hilarious in exploring some of these books. Like, for example, all the works of Shakespeare, except for the Hamlet, has been replaced with the word John. That's one of the books. It's one of all possible books. It's sort of, if you had a million monkeys typing all the time, you would get anything Shakespeare. This is the sort of version of that on steroids. And so what large language models are is a statistical model of the incredibly tiny fraction of that very high dimensional space, that extremely large space of possible books, that is the text generated by human beings. And we can build statistical models of that tiny fraction of that extremely high dimensional space. And it turns out to be very good at both recognizing and producing language that follows that tiny fraction of things that are sort of like what people would write. But the important thing when you're interacting with one or thinking about how they might be useful for you is that they are models of language. They are not minds themselves. They don't want anything. That's an easy way to tell whether something has a mind or not. Minds want things. They care about things. And these are just language models. And it's a little confusing because if you give an input to the language model that says, what do you want? It'll produce an answer. But that's just language. It's not a reflection of an actual desire. And furthermore, the texts they produce are statistical averages of what might come in response to anything before it, a prompt that would be in human text. So you get the kind of most likely outcomes, the most likely kind of following texts. And that's good for certain things, but some kinds of text, we don't want the most likely ones. I just read a paper this morning analyzing language model poetry, where likely outcomes are really fairly banal and dull. And what we really, when we read poetry and are moved by it, is about the special human connection we see in it. All right, that's enough about LLMs in general for the moment. One thing that is important about them is they really do have many good uses. And one thing that's been done a lot by AI people, particularly in science and medicine, is called information extraction. And the idea there is you take some text in some variety or another. So it might be a clinical note, it might be a journal article, but free text written by a human being. And we want to paraphrase it into a summary that's expressed in a structured vocabulary. It's a little like what Sam was talking about when he was talking about harmonizing data, except for this is kind of harmonizing language. And so this information extraction task has been widely used. We can generate the sort of harmonized summary of free text, and then we can do all kinds of things with that harmonized summary, take averages of that or whatever, do all kinds of things. There we go. All right. And what my lab has done in the last few years is sort of turn that on its head and say, okay, instead of doing information extraction, we're going to look at the biomedical literature and see if we can find texts that specify not information, but open questions or controversies or future directions that ought to be pursued, or many other kinds of statements that appear in these biomedical texts about knowledge we desire but don't yet have, things we think we could figure out soon. And it turns out those are really important. There's a lot of them. Almost every article published in the biomedical literature starts out with a set of questions described. Those are the questions the author thinks that they addressed in the article. And then towards the end of the article, you tend in the discussion section to get new questions. Well, these results raise the question of, or there's some controversy here. It seems different than previous results and the like. And we demonstrated in a paper that was published last year, and that you can see in the bottom left, that AI approaches using these language models and the like can identify pretty reliably statements of ignorance in the literature. And I thought I would pull as an example one of Doug's papers and pull out a statement of ignorance from one of Doug's papers. And I took a recent review of epilepsies and clinical management of them that he published. And it turns out he was surprised when he looked at it because it was actually his son who published this paper. Oops, sorry about that. But anyway, it's an interesting statement of ignorance. And you can kind of see that these are fairly complex. There's a lot of open questions that are referred to at the end of this survey article. And so what we're proposing to do, and we've been talking about this with CZI, is to use these statements of ignorance to try and find synergies in research on rare diseases. And let me pause for a moment and talk about why that's important. Because rare disease research is typically underfunded. There are a lot of rare diseases, depending on how you want to count, 7,000, maybe even 10,000. And so it's really not possible to put the kind of resources you would need to develop drugs and do the other kinds of work we'd like to see in 10,000 different diseases. So in order to address that problem, we need to collaborate. We need to find areas across particular diseases that we can use to make scarce resources go further. Now, that's been going on for a long time. A lot of the work that Sam has been doing and talking about the collaboratives and other kinds of issues that data commons raise is those collaborations are generally driven by shared phenotypes or maybe sometimes shared mechanisms. But what we're looking to do is to try and find shared questions, shared research areas that might lead to synergies that aren't identified by shared phenotypes or shared mechanisms. And our initial work was great in that we demonstrated we could do this. But what we did was go through journal articles and mark up ourselves what we thought the statements of ignorance were, which, if you're used to reading journal articles, it's not that hard to do. And then our programs were trained to identify particular sentences that had a statement of ignorance, or sometimes paragraphs, and then map those into those structured vocabularies. And that was great for demonstrating that it could be done, but it doesn't give us exactly the sort of shared questions that might lead to collaborations and synergies in research. So what we're proposing to do now, and CZI just announced in this round that they were going to fund this, so we're off to the races, is to try and create a more useful ignorance base. And we're focusing our work here on monogenic epilepsies. And so what we did previously to identify what counted as a statement of ignorance and the categories that we used to describe them were based on these sentences or paragraphs. And really the way we picked them was to be able to do it reproducibly. So multiple different human beings looking at those would pick out the same short passages and say they were the same category of ignorance. And that way we can train these machine learning methods to try and identify them. But that's really not what's going to drive the research synergies. So for the next year or so, what we want to be doing is talking to stakeholders across the monogenic epilepsies. And it doesn't have to be monogenic epilepsy. And it could be epilepsy adjacent, really anybody who wants to talk to us, and identify what they think are the important open questions and controversies and the like. And we want to do this at several different levels, right? So some of them, when we're talking to, say, parents, or caregivers, they're going to have different kinds of important open questions in their mind than maybe clinicians might have. And even clinicians are going to have different sort of open questions than more molecular researchers who are interested in trying to find mechanisms. And we'd like to be able to identify all of these different kinds of goals for new knowledge and then see how we can put them together to sort of relate them and find those synergies that we're looking for that might ultimately drive putting resources into those. And so what we'll do for the first year of this project is to get those interviews and surveys and then analyze them. And then in the second year of the project, we'll do another round of technical development so that we'll be able to use these AI techniques to generate more useful ignorance bases, ones that really reflect the needs of people living with these diseases, caring for people with these diseases, and researching the causes and new therapies for these diseases. And so I'm going to stop here so we have more time for a panel discussion. But I want to invite all of you who are listening to this to engage with us. We really want to talk to everybody, a widest possible variety of stakeholders in these disease areas. And we understand that you're all busy and have a lot to do. And so we have very flexible time commitments. If you're really gung-ho about this, we will do multiple Zoom-based interviews and really dig in on what you think the important questions are and the controversies and the things we need new knowledge about, and look at sort of the subtle details of those. Or if you're really busy, we will have a short survey that you can respond to in just a few minutes. We should be starting this work around the beginning of the new year, and I would encourage you to reach out to me at the email address in the slides. And with that, I will step aside and let us get to the panel discussion.
Great. That's great, Larry. Thank you so much. If Doug and Justin could come back on, and we can, I'm not sure, I can ask the organizers to make sure that we turn off the speaker view so that we can all see. And hopefully Justin can get his camera back on. There he is. All right. Appreciate all those talks. Obviously, there's been a lot of discussion in the chat, and we've been fielding a lot of questions. One of the things I just wanted to, and sorry, I'll invite people to submit more questions through the Q&A or in the chat if they'd like. One of the things I wanted to ask, and maybe this goes first to Justin, is What are the challenges that you think about in terms of data collection, both clinical surveys, EEGs? We've been hearing from so many groups, all the different ways they collect data, and it's almost dizzying the different ways that data come in. And I'm curious from your standpoint, if we're thinking of being a data coordinator or aggregator, what are the challenges you see and how should we be thinking about them?
I mean, I think one of the biggest problems, at least in our community, because our kids are so sick and the parents are so massively burdened, often it's a single parent doing the work, that just getting them to request the records and to even log into something that's a 5 question survey is challenging. I mean, how do you get through to a family member, a caregiver who's trying to keep seizures at bay and trying to manage reflux and changing diapers and healthy kids and doctor's visits and all these? It's a chaotic lifestyle. It is so unpredictable. And so I think our community in particular recognizes the importance of some of these tasks, but they just sometimes have a hard time doing it. And quite frankly, I'm deeply involved in all of these programs, and I sometimes have a hard time getting myself to do it because I am so chronically exhausted. So it's a major hurdle that the families kind of own, and we're constantly talking about how to incentivize families to do it the right way, and how do we avoid overburdening them, because we have surveys that go out through our registry, and then we ask them to participate as a citizen, and then we ask them, to go collect their EEGs, because that's raw EEG files kind of harder to get than connecting through citizens. So, unfortunately, I don't have answers. I see a lot of the very smart people that I'm friends with through other rare disease organization leaders who are here. Maybe they've come up with better solutions, but fighting the exhaustion part is tremendously challenging. They're just asked to do a lot of tasks. And so, from a foundation point of view, what we try to do, I guess, internally is space out the ask so that we don't get rejection. And we try to stratify it by saying, what's the single most important data point we need today and what's going to be most impactful? And the things that don't resonate as much end up falling off that long list of asks. So I don't know if that's much of an answer, but that's what we're fighting against, I think.
Have you dealt with issues around consent, especially as kids age into adulthood, and how have you thought about those issues?
You mean challenges getting patients to agree to consent to share because of the concerns they have about exposing their, the names or privacy issues, or?
Well, more like if a family gives you data from their child, say when they're 10, and you want to use it 10 years later for research, now you have data from somebody who's 20. There's obviously consent issues around having that patient consent. Have you had to deal with any of that?
Not that I can speak to directly. I mean, kids like Andrew are always going to be, legally under the guardianship of a parent like me. Andrew will never get to a point, well, without a meaningful, drug that rescues his phenotype, Andrew will never be giving consent. So we will be his proxies legally and otherwise. So we will always do, medical decision making. I don't know, for our more high functional AD and FLE phenotype, kids with nocturnal seizures, maybe that's an issue. But, I think I don't know how to address that because I have not had to tackle that very specific question.
Yeah, we're thinking about it obviously because we obviously want to get longitudinal data on kids and then young adults. So it's going to be an important issue. Okay, so I wanted to ask another question about just new tools, and maybe I'll start by asking Larry this question. We're all faced with new chatbots and LLMs and ChatGPT and all sorts of things, and we've all had experiences feeding questions into there. And how can the community think about these tools and how to use them responsibly in ways that can help them research their child's disease or research cures or clinical trials? How should we be thinking about that?
Well, that's a tough question. It really depends a lot on context. I think one of the best things to keep in mind is that you're dealing with a set of statistical averages. So if you're asking questions about something extraordinary, you should be very cautious about the results that you get. And if you're asking one of these chatbots something about something more normal or more common, then you're more likely to get an answer that is correct. For the time being, we're going to have to remain vigilant because one of the risks of these things is they produce really robust strongly supported looking answers that are some large percentage of the time correct and some not insignificant percentage of the time completely wrong. And they're very persuasive sounding even when they're wrong. So if you're using it for a high stakes decision, the answer is don't. For less than high stakes decisions, right? So you don't want large language models making decisions about launching nuclear weapons in life or death situations or any other kind of high stakes outcomes. On the other hand, they're a quick alternative to getting up to speed on some question that the answer is widely known on that looks a lot more specific to your question than trying to look it up in, I'm going to date myself now, say an encyclopedia.
Thanks, Larry. And I'm curious, both to Justin and Doug, how have you looked at these new tools in both your lives as general citizens and parents, and also as clinicians and researchers? How are you thinking about these? I'm sure lots of people are using them. Maybe Justin, you have perspective, and then Doug, maybe you have some ideas.
Yeah, I mean, I guess I've been reluctant to really delve into it from a medical perspective, but it certainly made, basic communication much faster and easier if we want to write a community update. So, I use it for things where, like Larry was kind of alluding to, these aren't critical decision, points. It's more just facilitating workflow than anything. And at some point, maybe we'll look into the utility from a medical perspective, but I haven't gone down that path yet.
Yeah, I agree, Sam. And in a conversation that Larry and I had, it was so interesting on this topic is that, Larry, as you explained to me, that these models could be good at predicting the next thing that you might say or completing a little portion of a picture that's missing, but not abstracting multiple points on a biologic, continuity of like, this enzyme does this, it causes this substrate to do this, which then does that. And so there's still a very important role in thinking through biologic situations for retaining control over that, if I understood our conversation correctly.
Yeah, Doug, I think you did. And that's one of the important things to note is even though they have the appearance of reasoning, they are not actually reasoning. So again, it's always language phenomena. So if you ask, explain to me how some long biological process works, explain TCA. There's a lot of explanations of TCA. It'll look really clear and straightforward, and it might really be helpful in getting people to understand it. On the other hand, that it's an appearance of reasoning, not actual reasoning. So it's not thinking through each step and putting them in collection. It's just reflecting what's in the literature. One thing that I will point out that is people currently have a lot of trust in them. And one of the things that has been good is getting people who are reticent to take in certain kinds of information, if they ask the chatbot about it, seem to be more likely to take it in than they would be otherwise. So if this is the best studies have been in looking at mis and disinformation in news. But nevertheless, rather than having an expert say, this is the truth, sometimes for some interlocutors, having an interaction with a chatbot is more persuasive to them than an interaction with a human expert.
I know that was really, I found that when we heard the presentation from the guy at Google to really be discouraging, that people found interacting with a chatbot more empathic sometimes than talking to their physician. Yeah.
Well, nobody has to get paid in 15-minute increments as a chatbot. Okay, they don't get paid at all. And so I think a lot of what you see, and in fact, that study, very famous study that showed that chatbots were not only as accurate as physicians, but came across as more empathetic, was done on a Reddit chat. The physicians who are answering those questions were doing it out of the goodness of their heart on their own time. Pajama time is sometimes called. And so maybe they were a little terse. Maybe they weren't quite as empathetic as a chatbot who has got who has no other demands on their time.
All right, before we leave this topic, Sam, I did think of one specific example that we've used it for. So for any of the parents out there who struggle with their IEPs and making sure that their schools build an IEP that's really best suited for them. Jason Curry from SCN2A on his website, he put together like a ChatGPT sort of module specifically to help you flesh out a really strong IEP. You just throw in the parts that are really critical to your child, and it'll sort of write itself for you, so that you're not signing off the one the school hands you. You're providing the document that you believe in to the school and letting them maybe edit as you see fit. So that's the only real world example I can really kind of give.
And Justin, you raise a really important point, which is that prompt engineering has become an incredible new science in and of itself. And actually using these tools effectively really comes down to how you write your prompt. So if you go on Reddit and other places, they will actually have prompts like one for the IEP or writing a white paper where you just fill in the blank like Mad Libs and it generates a really good response. So thank you for bringing that up. I wanted to ask this question about privacy because one of the most angry a patient ever got with me was when I didn't get consent from the parent to use the child's tumor before they went to the OR. And because of the way the consent worked, once the child was on the operating room table, I no longer was able to use consent to share that tissue. And the parents were so upset with me. I was a fellow at the time. And they just wanted their son's data and tissue to be used to help some other child. And I'm curious, Justin, what do you hear from parents about worries about privacy and security? Do you worry about your own child's, data being identified in a data set? And how should we be thinking about this tension, this push and pull between wanting to share, but also needing to maintain privacy and security?
Yeah, I mean, I'll speak to it, specifically. I mean, both as a patient and as the parent of a rare patient. I think that I think HIPAA is important. I think that we want to protect information. I think to some degree it's gone too far, like I alluded to in my slide deck. I think it's caused this cultural shift so that we are default no. By default we want to protect everything, and I think that creates an environment that's really challenging to get people to say, okay, we want to protect everything, but we also want to share. So for me, I mean, look, we're all posting our lives on Facebook, public facing platforms. I share everything about my kid on LinkedIn, Facebook, wherever I can, in the hopes that somebody will help us. And those tools become really helpful. In our private Facebook forums for our disease, parents share a tremendous amount of information. And we've, with their permission, we've brought in companies to harvest that data, because we get insights from that we don't get from our registry. Because you're answering questions, we're getting data that there wasn't a question made for, but we're getting data anyway. So, I'm a big proponent of encouraging our community to share to the degree that they're comfortable with. I try to give them the explanations for why I think it's important to share that data. I mean, at the end of the day, if you want to cure in a rare disease space, you have to be willing to give up everything, I think. And, for those who don't want anything, great, keep your information to yourself. But sometimes the greatest contribution for those who aren't leading their organizations and taking the time out of their way to do it, the single biggest contribution they can make is the willingness to share their data. So I preach that over and over. And for some patients or, parents and families, it resonates. And for some, it doesn't, so I don't know how to convert the ones for whom that message doesn't resonate, but I can sure, lead the ones for whom it does resonate.
Somebody made I'm interested also in what Larry and Doug think about this, but somebody made the comment to me that there's a huge difference between data sharing for cancer versus data sharing for a longitudinal chronic disease like monogenic epilepsy. Whereas if you share your data because you have cancer, you're sharing your data from a very discrete time point. We're not talking about survivorship or later effects. You're sharing your data for your diagnosis and your treatment and your outcome. Whereas if you're sharing your data for one of a monogenic epilepsy, you're basically sharing data from childhood out as long as you can go. And so maybe that makes people think differently about wanting to give up their data. I don't know if you've come across those kinds of attitudes. I think we're going to have to really be transparent with families as we take and use their data. Anyway, any thoughts about that? And to anyone?
Well, I think the transparency is the foundation for most of the work we're doing, right? It's how I treat my patients. I don't sell sunshine and rainbows to my patients when I'm seeing a breast cancer patient. I tell them the full spectrum of what to expect, good, bad, and otherwise. And what I tell them is my job is to educate them and empower them to make good decisions. If they hear that message and it scares them, then I am not the right doctor for them. But I counsel patients I would expect my family to be counseled so that they can be part of that decision making, right? We want to cut off the legs of paternalism and put some of the power in the patient's hand. And I expect that for our communities. You know, some people get scared by that, and that's okay. But I think that our jobs is to be transparent about what our motivations are, what are we going to do with the data, you know, and then give them some power and say in terms of maybe where that shared data goes. I think that people like the power of decision making, especially when you're stuck with a kid who you don't have the power to change yourself. Giving families some power, I think, is helpful towards recruiting them to the cause. I'll probably end there, otherwise I'm going to talk for 20 more minutes and drive people nuts.
No, you won't, Justin. But something came up when you were saying this, and you know that tool that we can use as physicians, where you can record a conversation with a patient, and then AI will turn it into a coherent note. Would that be something to get information that's valuable from patient families? You know, they, like you're saying, they don't have time to complete a survey, but maybe they have 5 minutes to talk about, you know, what's going on. Is that an opportunity, do you think?
I think our parents like sharing their stories, and I think the more simple the mechanism through which you're sharing that story, to me, the better. I can't speak to what kind of useful data comes out of that, but I think that our patients like having a voice, and if that becomes a reasonable methodology for harvesting what they want to share with us, then let's dig into it.
Well, I think the technology stack opens the door in some important ways. So there are plenty of people who are, you know, caregivers or parents of people with rare disease who are happy to type into Facebook or whatever and put up pictures and do that. But it also excludes a significant portion of the population that might have a much easier time sharing their stories if they could just talk about it rather than have to type it and organize it that way. So the new technology might definitely expand that opportunity to others. I do want to say something about HIPAA, and I very much share Justin's concerns, but one of the, I think, most grievous mistakes in that legislation was, while there are exceptions that allow hospitals and others to share patient data for business purposes, there are no exceptions that allow people to share data for research purposes. And that's a shame. And patients don't really generally understand that. And so making it clear that if they want their data or their child's data to be used for research to help others, they have to give permission for that. And I think often that's a surprise to people, and they want very much for their data to be used for research. There's a caveat to that. They worry about drug companies profiting off of it without making those drugs available to the patient community and in fair ways. And so there's a little bit of trade-off about who's doing the research and who benefits from it. But by and large, in general, people are surprised that their data cannot be used for research without explicit permission.
One thing that I thought of while you were talking was is the return of results. And we've had some parents tell us that they're frustrated that their data goes into this void and they don't know what happens to it. What kind of results do you think are important to return, and should we be telling patients your data were used in this paper or in this study, and what is the impact of that?
I think they do want to see something in return, right? When they spend minutes or hours or whatever the case may be providing this data, getting some insights back is important. That was one of the things that Citizen made really clear early on, is their goal to, take the data, analyze it, and share it back to the community, which is how we came up with numbers like 95% of our patients over 10 years old being on 5 drugs or more, even though Dr. Nordli and all his colleagues would say that our kids should never really be on ideally more than 2, maybe 3 at the same time, which means there's a tremendous opportunity to do better medicine today while we're waiting for the drugs of the future. And so we do try to share that back. I think we don't always share it back quickly enough. Sometimes efforts to get the data, there's the data compilation, then there's the analytics side, and sometimes we don't have the entire spectrum lined up, and sometimes there's delays. But I do think that we use it to encourage as a recruiting tool for the next effort to share back data from one program as we begin to ask them to share data for the next. So I do think most of them want to see something back. Treatment insights, especially, Dr. Nordli would probably suggest there's, of the 15 drugs that Andrew and others in our community have tried, there's probably 2, 3, or 4, maybe at most, that are probably the best drugs. And we've got data through Citizen and our KOLs who can sort of back that, up what the better regimens would be. So sharing that back to the community, it's instantly actionable, right? So these kids who are being managed in maybe not the highest end academic and tertiary centers for being, they're the only patient someone in the community is managing. It's great to give them, this data back from a larger cohort of patients to say, it's great that you're using these 2 drugs that you trained in residency on 25 years ago, but here's what the data from this compiled, amalgamation of patients who have KCNT1 really looks like. So I'll stop, but I think parents definitely want that feedback.
Yeah, that's helpful. I think we have time for one more quick question before hopefully Dr. Califf is going to get on at the top of the hour. That's what his folks said. He said when he would come on. We did get a question about the kinds of data that pharma companies are looking for drug development. And I know that there's some subtleties here in thinking about the kinds of data. I know that, Larry, you've been involved in a lot of work around pharmaceutical industry, and I'm curious what your thoughts about the kinds of data that pharma companies want, and certainly Doug and Justin, if you have thoughts as well.
Well, so pharma companies, starting from the very early preclinical phases, are interested in, first of all, finding a target that would affect, if we could modulate that target somehow, would affect the course of the disease. And so the first kind of information is what are the causal molecular mechanisms involved in this phenotype to the degree that we can understand them. And so a lot of the data that's generated is toward trying to understand that molecular cause. In addition to having at least some hypothesis about what the molecular causes are, what's the gene, what's the interactions, what are the regulatory aspects, we're also interested in good models. So in order to be able to do preclinical work, we have to have either in vitro, test tube assays of, does this, can we affect that target? In animals, do we have some good animal model that we could tell whether or not we're affecting the phenotype or whatever it is we're trying to modulate that target to do? And then we also need safety data around it. What else does that target do? If we modulate that target, what else will change? And those are the 3 kind of main preclinical questions that pharma comes to the table with. What are the causes? What kind of models do we have that we can explore possible treatments with? And what are some of the safety issues that come up when we modulate those targets?
We've been getting a fair amount of external questions for our pediatric cancer data around using data for external controls for clinical trials. And now that we've collected data on almost 50,000 kids with cancer, I think this is going to be an important issue for running trials if you have a set of matched controls to run. So I'm hoping that is an important area going forward. Justin, have groups approached you and your groups about I don't want to say monetizing, but about leveraging data for drug development, and how have you approached that?
Yeah, we certainly have had those interactions. I think the way we try to figure out how to, if we can use it as an incentive tool to get parents to participate and pharma to take an interest in us, then I think that, we win. At the end of the day, we want to have, like I kind of referred to, this sort of community garden where anybody can come in and do research. They can come in and do drug discovery because we have this asset pool. And so, if somebody's monetizing it, well, that's fine. It keeps them interested in us as a disease. Anything that can promote the accumulation and distribution of our data ensures that pharma is going to be interested in us, because pharma wants to go after diseases that have databases. You can't just randomly pick a disease and then hope for the best. You want to go after the ones that are well organized, that have data available, so that ultimately, if they're going to submit an IND, they've got the data to go along with that drug that they think might work. I don't know if that answers your question, but it's what came to mind.
Have you heard any, Doug, you may have thoughts about this too, but have you heard any thoughts from patient groups about doing their own drug development? When I was at the Science and Society meeting at the Chan Zuckerberg 2 weeks ago, there was a group there that's actually thinking about acting almost as their own CRO and doing their own drug development. Have you come across, any of you have ever heard of that?
Yeah, absolutely. And Sam, do you remember that presentation at CZI where one of the groups is actually doing that. They're funding their own lab, and they're having their investigators working out of this. I think it's a shared space in another organization, but it's their lab, and they have reagents and everything, so that when they have a question, they can hit the ground running. They don't have to wait for the whole buildup. So yeah, definitely have heard that. And I mean, it's remarkable what groups have done, like developing, their own, helping to fund or get people interested in developing animal models and figuring out ways to develop ASOs. So it's been pretty incredible to watch. From just a quick thing about when I'm contacted by pharma, which is not that frequently, but when I am, their typical question to me is, Doug, what is the EEG biomarker? So I think that is of interest to them, and that was another reason that we selected this as an important thing to have in a central repository.
Sam, I'll answer two questions for you. One, you said, what's the data the pharma is looking for? When they can approach us, they ask questions like, how many patients do we have in our community, both in the United States and worldwide? Where are they? What hospitals are they being treated at? And then who's actually interested in trials? And then break it down by who's interested in small molecule versus ASO, because it's different types of patient, different decision making in terms of participating in those. They ask us how aware are we of what level of risk patients in our community will take for a drug? Would they take the risk of potential death, which we've seen in rare disease drug trials? What's the quality of life of our community, and what sort of comorbidities do our patients have? I think in terms of your other question, yeah, I mean, unfortunately, and there's people, I think, friends of mine in this, who are on this, who have logged in today, who are really take the reins for pushing drug discovery, right? So, and I see Dr. Califf, I'll get off quickly, but the bottom line is, we've run our own repurposing program, and we've initiated, de novo small molecule programs, ASOs, siRNA, and editing, because if we sit back and wait, our chances of success are low. We had a few de novo programs that started for KCNT1, but if you look at that math formula, 90% of those are going to fail. So I told our community, it's great that they're interested, but we need 10 programs to ensure that there's one potential real program for us. So, and a lot of this is really driven by us reaching out to pharma and initiating, and in some cases funding these programs.
Thanks, Justin. And thanks, Larry and Doug. Wonderful. So glad you could join, Dr. Califf. We've spent the last 2 hours talking about rare disease research and talking about monogenic epilepsies. Justin is a physician and a parent, and we've had many parent groups on talking about how to share data that can be then leveraged for drug development. And we'd love it if you could just spend a few minutes talking about your perspectives. I think most people know you, Dr. Califf. You're the current FDA chair and was also chair back in 2016 as the 22nd commissioner. You were at Duke for a long time running clinical and translational research. And actually, I met you, Dr. Califf, through our work through our CTSA grant and through the CR Forum, and I've enjoyed knowing you over the past 15 years or so. So we're so appreciative of you being here today. I know your schedule is packed, and look forward to some comments if you can make about how we should be thinking about rare disease drug development. Thank you.
Well, thanks, Sam. I'm coming into this obviously relatively cold, not having heard what you've talked about for the last 2 hours, although Justin's comments, I think, hit a very important point I would have always made, which is no matter how smart we think we are about biology, it's still the case today that about 90% of drugs that get into phase 1 clinical trials don't make it to market because they either don't hit the target the way it was expected, they have an unexpected toxicity, or they hit the target, but the integrative biology doesn't lead to a clinical effect that where the benefits outweigh the risks. So it is a tough game, even as we're seeing this explosion in knowledge that is improving our capability quite a bit. So the main, maybe just to make a few quick points, and then I'm happy just to have a discussion. That would probably be best for me. It's been quite a day already today, and more to come. But what I'd say is this is, as you pointed out, I've been working in clinical and translational research for almost 50 years now. And much of what's happening today would be in the category of things we only dreamed about 30 years ago. And as we saw the Human Genome Project and integrative systems biology come into play and the computational capacity, all the pieces are falling into place now where it does seem entirely plausible that many of the rare diseases, particularly those with their genetic basis, are going to be approachable with treatments that interventions that are effective, at least to some extent, and for some even curative. And so it's a great time to be excited. We've responded at FDA now by creating what we call the Rare Disease Hub, because what we were finding was that we have drugs and biologics, and often in approaches to the same disease, different interventions might be used that would fall into either category with almost exactly the same biomarkers and targets. And so it didn't make sense to be giving two different sets of advice. And there are many common methodologic issues which need a lot of work, particularly in how we quantify probabilities of benefit outweighing risk in cases where we don't have enough individuals in the clinical trials to do an empirical calculation of risk and benefit. And so the components of it, which I understand, Sam, you're particularly focused on, I would say that sharing data becomes absolutely essential and on multiple fronts. On the one hand, having competition is good in most places, but when you have a rare disease and limited capital, you really can't afford to have people come up short just because they couldn't get the firepower it takes, remembering that you not only have to have a target and you have to have a intervention, whether it's gene editing or small molecule change of gene function or a traditional approach to functional or symptomatic component of a rare disease. You have to have an intervention that can not only work, but also can be manufactured in a reliable way and eventually at scale, because the distribution system becomes very important. So the financial needs are significant, and collaboration is a much better approach. And I think we've seen that in a number of rare diseases now, where you had individuals working separately, and then if they had a compelling force to work together, the end product was much more achievable, and the results were better. But then on the clinical front, simply understanding what the trajectory of the disease is so important in situations where we can't do a traditional randomized clinical trial. And we've had, just in the last year, some cases where the variability in disease outcomes is greater than what was observed in the trial, and it's led to very much difficulty deciding what to do in the review for marketing status. So we would love to see a lot more registries of diseases in which people were sharing their data as individuals and as health systems into composite pictures of characteristics of people and what their outcomes are over time as a backdrop. And then we have to think about the evidence for that an intervention is effective. And there are multiple methods that might be used, but fundamentally, they're all dependent on having the biggest sample size possible in the case of rare diseases, to have the most confidence that whatever inference that we're making from the data actually will hold up when put into the practice if the product gets on the market. And then I can't talk about this without also mentioning the diagnostic odyssey. And this has been a very contentious issue, particularly with academic medical centers, but I've got some relatives going through, it's not rare disease, but cancer issues now, and the variation in laboratory testing, it's just not good for patients. And so it's really important that those developing laboratory tests collaborate so that we have enough people undergoing the tests to understand what the tests are actually telling us, so that we can have confidence that if we say we're identifying a person with X disease or X manifestations of that disease, that that's actually what's happening. Beyond that, we are seeing a number of failures because of lack of attention to the critical issues in biomarker assays. And, you know, it's understandable. I'm a longtime academic, so traditional academics like to make the discovery and move on. But knowing that moving a biomarker has a relationship to a disease outcome in concept is different than being able to reliably measure that biomarker when it gets out of the individual's lab that worked on it. And we're seeing a lot of cases where that just didn't get enough attention. And if you can't be certain that what you're measuring is accurate, you got a real hard time making decisions in cases of accelerated approval where you're not depending on clinical outcomes, but you're depending on change in a biomarker that may ultimately, with enough data, end up being a valid surrogate. But in the case of rare diseases, almost 100% of the accelerated approvals are based on biomarkers which are not proven surrogates, but are reasonably likely to be valid measures of disease status where a change in the biomarker will predict a change in the disease outcome. All of this is dependent on sharing data. And of course, Sam, you know a lot more about this than I do, but I did have 5 years at Alphabet and a lot of time in academia working on these things. And I think it's fair to say that there are no technical issues now in sharing the data that we need to share. The issues are really artifacts of culture and boundaries that have been set up for reasons that I think are unfortunate, but something that we're going to need to continue to work on. And then finally, this is not just a U.S. issue, and it's don't have to be a mathematical genius to know that the 340 million people that live in the U.S. are less than 4% of the world's population. So if you have a disease that's rare, imagine if you had access to all of the 8 billion people in the world, and those individuals who had a rare genetic disease, you'd have a much greater ability to rationally develop effective interventions. So I'll stop there. I'm glad you guys are meeting about these kinds of issues. And I guess the last thing I'll say is I've learned a lot over the years about advocacy groups, and I think it's just empirically a fact to say that if you have a good advocacy group that understands how this all works, the chances that your disease of interest is going to eventually have an effective treatment is just much higher. And so I worry a lot about, I mean, one reason to have these kinds of meetings and to keep talking is I worry a lot about people with diseases who are not well represented by advocates who can raise money and push the right things and get the feel for that disease where it needs to be. I'll stop there. Glad to take a few questions or hear comments if you
Thanks,
have them.
Dr. Califf. Yeah, we won't take too much of your time, and we are interested in this long tail of rare disease and the advocacy groups that are working with them. Chan Zuckerberg has been, very gracious in their funding of through their Rare as One network and funding rare disease initiatives. One of the things that's come up is understanding how groups can take their data and let pharma companies leverage it. And we always, especially in pediatric cancer, we get a little bit cautious when somebody wants to buy our data. And I'm curious from your perspective, has the environment changed in such that we should be thinking about more ways to get data into the hands of pharma companies that can develop? What are the gotchas there that we should be mindful of?
I mean, I think it's obvious that you can't develop, gain regulatory approval, manufacture and distribute a drug without having a company. That just doesn't work. And so, when you say pharma, if you mean a company that manufactures and distributes a drug or a biologic, I don't know how you get around involvement, but I would say this is where, there's a very strong view at FDA that product development should be patient-centered, and that advocates or representatives of the patient population should be involved at every step of the way, the design of clinical trials, et cetera. Obviously, not so much in the manufacturing process, which you wouldn't expect patient advocates to be involved in that, but certainly the clinical development program should be something that is designed with the patients. And we've pushed for that across the spectrum of medical products as a preference. We can't force people to think that way, but I think that's happening more often. So from my view, the best safeguard for the patient group is to be involved. And I'll just point to cystic fibrosis and type 1 diabetes as areas that this has been written about. But if you're well enough organized, you really have a lot of power as a patient group. Also would throw clinicians into that mix. One thing I'm very worried about right now for rare disease, we were descended upon by a group of senior pediatricians a couple of months ago. I didn't know what to expect. There are a lot of interesting issues, but what they wanted to say was, no one's going into pediatrics now. And the pressure on pediatricians to just churn through the clinical mill, just like my adult colleagues I've heard so much from since I stopped practice to come to FDA. Who's going to do these trials? And I was very involved in the Pompe's disease developments because they happened at Duke where I was. And it's also the case, as I mentioned, that in many cases, the treatment's not going to be a cure. What it's going to do is allow people to live a lot longer, but they often will have a chronic disease, which is previously undescribed because no one was alive with the basic genetic issue, to grow into adulthood. There, I feel like a personal expert and an advocate because I have a daughter who had really serious congenital heart disease of the type where no one would have survived. But in 1978, she had a huge operation, and now she's in her 40s, doing well, but she's not, she requires more medical attention than someone who's perfectly healthy in her 40s. So I think when you talk about advocacy, we got to throw the relevant disease specific clinicians who are driven to care for people with these problems.
Yeah, it's interesting you said that. When I was in Minneapolis 2 weeks ago, I talked to somebody about building a data commons for adults with congenital heart disease, just to provide all their longitudinal data in a way that could be followed and studied. So as you know, we don't follow survivorship well, and we don't track patients over time through multiple institutions very well. And to your point, technology is solved. These are social human issues, and we need to overcome those as well. I know Dr. Hunter, Larry had a quick question, and I want to let him ask, and then we'll probably be just about out of time by then. But hopefully, Larry can ask his question real quick.
All right. Well, Commissioner, thank you very much for coming. And my question is really about the use of real world data in rare disease. So there are so few patients, and when there is a promising compound, especially for repurposing, but for other purposes as well, patients and their caregivers clearly want them. And so putting together control groups who don't get the intervention in the traditional clinical trial model is difficult. And there have been recently attempts to use characterizations, the natural history of the disease, to try and, use self-controls, as it were. And so FDA has been receptive to using that sort of real world data. And I wonder if you might tell us a little bit about the standards for quality or other kinds of considerations that go into assembling really good real world data sets that FDA could use.
Well, as you may know, this has been a big part of my career, so I'll try to keep it brief. I would just say there are a number of guidances that have been published, and more to come out about data quality. And I'd say in the case of an individual disease and an individual application, there are principles. The first one is you should just pay attention to the quality of the data, which unfortunately too many people just say, I've got a bunch of data. Let's just pardon me, but there was an old saying I learned in North Carolina, you can't make chicken salad out of chicken shit. That's an old statement. So I think purposeful paying attention to quality of the data. And one of the things that's most important is continuity of follow-up, which Sam referred to. It's a general problem. And I would say it's a truism of clinical epidemiology that the most important information is in the people that you lose to follow-up, either because they died and you didn't know it, or something bad happened and they ended up going to another health system and you don't have a record of it. And so part of the intentionality for useful real-world data is getting complete follow-up. And Sam, I'm still astounded. I was in a meeting yesterday where we're estimating now and we're doing better, but a lot of places still don't know who's dead and alive in their systems, which seems like principle number one.
And we're paying big money for the Social Security Death Index so that we can do that externally. It's a huge problem.
Yeah. And so one of the fastest growing areas, obviously, in the real world data arena is sensor data and other information coming in collected by the patients themselves. And I think everybody at FDA has great hopes for that, but it still has all the same methodologic issues that need to be solved, particularly around things like missingness, artifact. I learned a lot about this during my time at Google. I would say every day I had some software engineer who called me up and say, I got a fix for this or that health problem. And you could easily construct a slide that had like, here's how it should work in theory. But then you put it in the real world and it's often very hard the most important information may be missing because when things happen to people, they don't record it. And artifact is a real issue, but these are all solvable problems. That's the good news. So I think the intentionality of just paying attention to the, and I'd refer you to the guidances about the attributes of the quality of data. Amy Abernathy did a lot of this when she was at FDA, and I think had a lot of knowledge about it. So I think people at FDA would love it if we get more and more high quality real world data.
That's great. Thanks, Dr. Califf. Yeah, I think that's been our impression too. We're actually involved in developing some direct to patient tools now that will allow us to use the APIs that the EHR vendors have to pull the data straight out of the EHRs and dealing directly with the patients instead of the institutions. And obviously there's a lot of issues around the Part 11 compliance and how those data can be used for downstream applications. But I think, as you said, the technology is moving so fast, we just need to figure out the best ways to leverage it at the highest quality.
I would turn your attention to something called Home as a Healthcare Hub, which is a pilot project done within FDA. It's been announced and the prototype is done, I'm told, and will soon be available for people to look at. But the concept here is we want to channel industry into what you said, Sam, which is um, creating a composite of tools and apps that help people live at home and monitor their health. And one of the problems we're seeing, I always refer to the glass cockpit project in aviation, where it used to be that when engineers could measure something new, they'd just stick a gauge in the cockpit in some random place. And the pilots couldn't fly the planes after a while because they didn't know what to look at and how to integrate the information. And I would argue that both patients and clinicians right now are in that situation where we got multiple vendor-driven applications that would be useful if they were integrated into a knowledge base that a human being could actually use. And so we're hoping to create prototypes that will help the industry move in that direction. And I may be biased because of my clinical history, but again, I want to argue that either just catering to the clinicians or just to the patients at home, either of those is a mistake. What we need are serving both and then connecting the two, because what we're seeing on the bigger scale is that people don't want to live in a purely virtual world clinically. They want a human being to deal with, but they sure would love it if that human being was assisted in being a helper in the clinical interaction.
Are you concerned, Dr. Califf, that the consumer devices run black box algorithms that tell you how many steps you took or what your beat to beat heart rate variability is based on the accelerometer and other things in there? I wonder if is that an emerging problem in how we assess the quality of these data, since we can't often evaluate the algorithms underneath them?
Well, I'm actually not allowed to talk about that as long as they're not advertising that it diagnoses or treats a disease.
Yeah.
And so, but once you do that, it's essential that, well, the FDA, it's not a black box if it comes to the FDA. We can look inside the box, and but it does, the trouble area there, which I experience every day at Google too. Once you've got like a device which is not to treat or diagnose a disease, but you can begin to see the application could be used for that, and it's in wide scale use. That's an area of technology where I think we have a lot of work to do, but there's some responsible actors who I think have done a good job of saying, I'll just point to Apple Watch. Since I used to work at Google, I'll point to Apple. When they got their atrial fib indication, they had to actually show what was in the algorithm and do the clinical studies. And so I think there is a way to do that. Now, decision support, that's a whole other issue, which I'm very worried about that, because a lot of that's being done as practice of medicine, and it's not even in the FDA's regulatory scope right now. But you probably, if you're like me, when, like I'm going to get in my car and this afternoon I'm going to say, I want to go home from White Oak, and it's going to tell me exactly where to go to avoid this brutal Washington I-95 traffic, and I'm going to take it at its word. I think clinicians are going to do the same thing. So if someone's not looking at the operating characteristics of those decision support algorithms, there could be some real bad things that could happen.
Yeah, and we don't need to spend the next half hour talking about medical education and data science and informatics, but I do think we need to train this new generation of clinicians as to how to evaluate these tools and how to use them responsibly. So I really appreciate you saying that.
All right. Well, thanks for giving me a chance to interact with you guys, and good luck.
Thanks, Dr. Califf. We really appreciate it.
It is a great day. We just got a few problems to solve along the way.
We know. Good luck out there. Be careful. Thanks, Dr. Califf. Thanks, everybody else. Appreciate it. Thanks for coming, and we'll be in touch. Thanks, everybody. Bye-bye.